Hello everyone, in this video, we will discuss about colony forming unit and calculation of CFU per ml. Colony forming unit or CFU. Colony forming unit is a unit of viable bacterial or fungal cells. Let's understand it with the help of an example. Suppose 1 ml bacterial suspension contains 9 viable bacterial cells and 2 dead bacterial cells. To know how many viable bacterial cells are present in bacterial suspension, we plated 1 ml bacterial suspension on agar plate. As viable bacterial cells are multiplied by binary fission, bacterial colonies will appear on agar plate. By counting these bacterial colonies, we can know viable bacterial cells in initial bacterial suspension. Importance of CFU. CFU gives us information about viable bacterial concentration of bacterial suspension. Let's understand it with the help of an example. There are 2 bacterial suspension of 0.0010 D. 1 bacterial suspension contains 14 viable bacterial cells and 2 dead bacterial cells. When we inoculated this bacterial suspension in plant showed chlorosis at 3-day post-inoculation. Another bacterial suspension contains 2 viable bacterial cells and 14 dead bacterial cells. When we inoculated this bacterial suspension in plants, there was no effect on plant at 3-day post-inoculation. Although OD of 2 bacterial suspension is 0.001, but due to difference in viable bacterial cells, 1 bacterial suspension showed symptom while 2nd bacterial suspension did not show any effect. We can measure viable bacterial concentration in CFU per ml. Calculation of CFU per ml. We can calculate CFU per ml by applying one of these formula. For example, 1 ml bacterial suspension contains 10 viable bacterial cells. We diluted it 10 times and plated it on agar plate, and we found 1 colony on plate. So here number of colonies 1, volume of original bacterial suspension 1 ml or 1000 microliter. Dilution factor 10. By putting these values in above formula, we can know viable bacterial count in bacterial suspension. Let's discuss another example. Suppose 1 ml bacterial suspension contains 1000 viable bacterial cells. We did serial dilution and diluted it 100 times. 100 microliter of 100 times diluted bacterial suspension plated on agar plate. So here colony on plate 1, volume of original culture 1 ml, dilution factor 100, volume plated 0.1 ml or 100 microliter. By putting these values in above discussed formula, we can know viable bacterial cells in initial bacterial suspension. Thanks for watching this video.